All right, folks, so it turns out I had a little bit of trouble with the last two videos, so I'm going to have to start the last two again. Let's pick up where we left off. So several things that Mendel discovered when he did his monohybrid crosses was that we had these traits that were either dominant or recessive. You saw the trait or you didn't, depending on the combinations that were put together in the offspring. So the terms that we're going to be using are as follows. The factors that Mendel was talking about, we're now calling genes. At the time, he just called them factors or genetic factors. Now we know that they're genes and that they're distinct sequences in DNA. Alleles are different versions of the same gene. And the way I think of alleles is like genes, a gene would be like ice cream, and alleles are the different flavors. So ice cream is ice cream, right? And you can have vanilla, chocolate, or strawberry. Those would be alleles of ice cream. Each individual in an either-or situation would have two alleles of a given gene. Now, as we go through this course, you'll see that there are sometimes more than two alleles for a given gene. But let's say for right now that you only have two alleles, one that would be dominant and one that's recessive. You see it or you don't. An individual that has two identical alleles is homozygous, homo for the same and zygous for occurring. An individual with two different alleles is heterozygous, hetero for opposite. And so when we're dealing with determining whether or not an individual is homozygous or heterozygous, what we're referring to is what we call the genotype. The genotype are the composition of alleles or the composition of individual elements that make up the pattern seen in the individual. The phenotype is what you physically see. So genotype would be the genes or the alleles, and phenotype is what you physically see. And so in this example here, what you would see here in the P generation or the parental generation, the genotype is big T, or homozygous dominant, but the phenotype is a tall plant. Over here for this plant, it's little t, or homozygous recessive, and the phenotype would be dwarf plant. In the F1 or the first filial generation, you're seeing a heterozygous individual, big T, and the plant is tall, so that's the phenotype. So what you're looking at here is the parental generation, F1 or first filial generation, and the F2 generation. Now all of this mixing and hybridization probably was pretty hard to track, quite frankly, until the 1900s came about, when Reginald Punnett, a geneticist in the early 1900s, came up with kind of a shorthand pattern grid. It was a technique that he used to show how two organisms could be hybridized, and you could predict the outcome of that cross. And we still call that technique a Punnett Square. And so to do a Punnett Square, what you're going to do is you're going to start what I would do. Let's come down here. I would first make a square and break it up into 4 quadrants. Then you write down the genotypes of the parents. So again, the genotype is the allelic composition of each parent. The male parent is always going to have the genotype running across the top of the quadrants, and the female genotype is going to run down the side. All right, so you get the male and the female genotypes, okay, which in this case are both heterozygous. The gametes separately are big T, little t, and then you fill in the genotypes of the offspring associated with each quadrant. So for this gamete and this gamete, when the male and the female gametes come together, you'll see that this individual you would predict would be a homozygous dominant. These two would be heterozygous, and this one is homozygous recessive. And this is all for a single character. You determine the relative proportions for each genotype and for each phenotype. So here you have homozygous dominant, homozygous recessive, each being 1, and then 2 heterozygotes. This is what we call the genotypic ratio, the 1 to 2 to 1 classic ratio of a monohybrid cross where both parents are heterozygous. I sometimes make a mistake on that. The phenotypic ratio, though, is 3 tall to 1 dwarf. So what you're doing is you're looking at the allelic composition here, or the appearance. So you need to be sure when it comes to the time to quiz time or test time that you understand the difference between genotypic and phenotypic ratios. In Mendel's experiments, he produced many individual crosses, monohybrid crosses, but then he started realizing that, well, maybe I could track two traits at the same time. And this is what we call a dihybrid cross. In addition to kind of making the whole process go a little faster because you're tracking two traits at one time, you can also ask the question, does one trait depend on the other? So for example, in humans, do people with blonde hair always have blue eyes? Or are those patterns of inheritance just coincidental? And so in this case, what Mendel did was he looked at character number 1, seed texture. Was the seed round or was it wrinkled? And character number 2, the seed color, yellow versus green. And so this is kind of a very difficult slide to look at. I encourage you to look at it yourself, but let's move over here to how to set up a dihybrid cross. Then we can go back to that. So when you're setting up a dihybrid cross, instead of setting up a Punnett square that has only 4 quadrants, you're going to set up a Punnett square that has 4 rows of 4. So you have 16 quadrants. Again, you're going to be looking at trait and color. So big R would be round and little r would be wrinkled. Big Y would be yellow, little y would be green. In this particular case, this individual, this parent plant, is producing seeds that are round and yellow, and this parent plant is producing wrinkled and yellow. And so, wrinkled and green. You want to look at the gametes that each of these form. Now, what's hard about this one? If you were to, let's say, for example, try to figure out how many different combinations of gametes that this particular plant is producing, you only have one option, big R, big Y, because big R and big Y are your only allelic compositions that you have here. A simpler way of determining this is using the old FOIL method that you did in math. So to determine how many combinations you have here, you would, in the 2 parental individuals, you would be first by first, big R, big Y, outer by outer, inner by inner, last by last. Remember FOIL from math? You know, you do first, outer, inner, last. You do that cross, which you're not seeing here, and you end up with all individuals being heterozygous in the F1 generation. Now figure out the gamete possibilities for this. Big R, big Y for first by first. Here's big R, big Y. Big R, little y, outer by outer. Big R, little y. Little r, big Y. Little r, big Y. That's inner by inner, and then last by last, little r, little y. Okay? And now you follow each of these combinations down into the square. Always keep your r's and y's consistent. So big R, big Y, big Y. Big R, here it is, big R, big Y, little y. The way I always do this is I always put the capital first, but I keep the r's and the y's together. When you look at this particular analysis, what you're going to find is a 9:3 to 3:1 phenotypic ratio. Okay, the genotypic ratio is a little bit different, but the phenotypic ratio, 9:3 to 3:1, is a classic phenotypic ratio that Mendel described as a consequence of an F1 plant that was heterozygous for both traits. So here's the data. 350, then these are actually Mendel's data. 315 round yellow seeds, 101 wrinkled yellow seeds, 108 round green seeds, 32 green wrinkled seeds. So how do we interpret these data? Well, it turns out that round and green and wrinkled and yellow in the F2 generation generated new combinations. Because remember in the beginning, the parents were round and yellow, wrinkled and green. And by the end, you had wrinkled and yellow and round and green, okay? And that was totally new. Those are called recombinant organisms. And so, or you can also call them non-parental organisms. And so these non-parentals would be predicted by using this Punnett square with this theoretical analysis. Fortunately for us, Mendel did that in the very beginning, so it's a lot easier to interpret nowadays. But the phenotypic ratio of the F2 generation, the 9:3 to 3:1, always ends up occurring if the genes are independent of one another. In other words, the wrinkliness and the color, seed texture and color, are not linked. In subsequent chapters in this book, we will be talking about genes that are linked. In other words, does blonde hair and blue eyes go together? Not necessarily. A certain percentage of the time they do. Here, round and wrinkled don't travel together. So we call this the law of independent assortment. So during gamete formation, any pairs of genes can separate independently of one another. In other words, seed texture does not rely on seed color. And so monohybrid crosses and dihybrid crosses are usually presented in diagrams that are a lot easier to read than this thing. This is much more of what Mendel was actually doing. Here are the possible male gametes or the possible female gametes. Look at all those arrows. Ugh, what a headache. This is a lot tidier here. This is showing you tall yellow. In this case, they're using the height of the plant. Tall yellow or short, dwarf green. Here is your genotypic ratio, 1 to 2 to 4 to 2 to 1 to 2 to 1 to 2 to 1. Or the phenotypic ratio, 9 to 3 to 3 to 1. If you end up with the 9 to 3 to 3 to 1 ratio, whenever you do a dihybrid cross, you know that those two genes are traveling independently of one another. The expression does not rely on the other. Now, an easier way of doing this is by just isolating a single row. I'm not going to bug you about this. Independent assortment typically can also be shown by using what we call a dihybrid test cross. For now, I don't want you guys to focus on this too much, but I do want you to understand that a test cross, when you do a test cross, you're always using one of the two plants or one of the two individuals is going to be the homozygous recessive for both traits. So test crosses always use homozygous recessive individuals. That's all I need you to know for right now. So let's move on a little bit in terms of modern genetics. So modern genetics now uses additional approaches to try to understand the expression of genes at different levels. What do I mean? When Mendel was able to do this type of work, all he could see was the outside of the plant, the flower, the height, the seed shape, the seed color. Based on that, he could just say, well, factors that influence the appearance of this trait are there or they're not there. Nowadays, using microscopes and molecular biology, we can actually look at, is the gene present, and if it is it being expressed appropriately? And if it's being expressed, but you have a mutation that occurs, maybe there's something else affecting the expression of that gene. So it helps us to identify an individual with defective genes, defective alleles, and it also allows us to look at how that type of defect is transmitted across generations. And so a lot of times when you see defective genes or defective alleles in organisms, we now call those loss of function alleles. It turns out that a lot of what Mendel was looking at were loss of functional alleles, especially color. When you have the absence of a color, it doesn't necessarily mean that, let's say in white flowers, that purple isn't necessarily, I'm sorry, I should say when you have a purple flower, it doesn't mean that white's not there. It just means that purple is masking the white. All right, we're going to stop here, and I will pick up with the chromosomal theory of inheritance in the next video.